G'day everyone, and welcome to PHY 2032. My name's Craig Harrison. There are approximately 40 trillion cells in the human body, double that if you include the non-human cells that comprise the microbiome, and all of these cells must continually perform work to stay alive. This work includes the maintenance of cellular composition and structural integrity, together with the integrated synthesis and breakdown of macromolecules and organelles. Sometimes cells work together, such as the contraction of muscle fibers of the diaphragm, to support the body as a whole. On average, the resting metabolic rate of a relaxed, awake, stationary, healthy adult human accounts for about 70% of the total energy expenditure each day. But additional work is required of cells when the body is either 1. Engaged in physical activities such as manual labour, exercise or outdoor play, 2. Undergoing periods of rapid growth such as puberty, pregnancy and breastfeeding, or 3. Fighting a serious illness or healing damaged tissues. To perform this work, cells need fuels, together with the capacity to convert these fuels into potential chemical energy in the form of ATP. Cells then convert the energy within ATP into chemical and mechanical work. This means that the need for ATP is immediate and unending, and consequently all cells must continually synthesize ATP, but this places a demand on the body to continually supply fuel in some form to all cells. Now, all fuels originate from the diet, but humans do not eat continuously throughout the day. Thus, the constant cellular demand for fuels to make ATP and perform work is paired with an intermittent intake of fuels. Those of you who have done some biochemistry previously will know that diet-derived glucose, fats and proteins are oxidized for ATP, but some are also stored for future use, and some are converted to other fuels that can be used by other cell types, depending on the metabolic phase, type of fuel and cell type in question. By metabolic phase, I mean the hourly and daily differences in fuel usage and energy metabolism, which are dictated largely by the abundance or scarcity of certain fuels and orchestrated, as we'll see in the next few videos, by phase-specific hormones. In general, there are two major metabolic phases the body undergoes in providing energy for cellular activities. These are the absorptive phase, during which ingested nutrients enter the blood from the gastrointestinal tract, and the post-absorptive phase, during which the gastrointestinal tract is empty of nutrients and the body's own stores must supply energy. Because an average meal requires approximately 4 hours for complete absorption, our usual 3-meal-a-day pattern places us in the post-absorptive phase during the late morning, again in the late afternoon, and during most of the night. Over the next few slides, we will examine the major metabolic pathways of the absorptive and post-absorptive phases, and then I'll summarise these pathways at the conclusion of this video. Now, in weeks 7 and 8 of this unit, Associate Professor Julia Choate will describe the digestion of carbohydrates, fats and proteins, but for our purposes we will start with the breakdown products of these fuels about to enter the blood or lymph during the absorptive phase. First, all cells take up amino acids for protein synthesis. However, liver cells also use some amino acids to generate carbohydrate intermediates, known as alpha-keto acids, by removal of the amino group. Alpha-keto acids can enter the Krebs cycle to provide energy for liver cells. Absorbed lipids are packaged into chylomicrons that enter the lymph and from there the circulation. The fatty acids of plasma chylomicrons are released mainly within adipose tissue capillaries by the action of endothelial lipoprotein lipase. The released fatty acids then diffuse into adipocytes and combine with glycerol 3-phosphate, which itself is synthesized in adipocytes from glucose metabolites, to form triglycerides. Glucose is the body's major energy source during the absorptive state. Much of the absorbed glucose enters cells and is catabolized to carbon dioxide and water, in the process releasing energy that is used for ATP formation. Glucose can also be converted to glycogen in liver and muscle, and glycerol 3-phosphate and fatty acids, the precursors of triglycerides, in adipose tissue and liver. The major function of the post-absorptive stage is to ensure that blood glucose levels are maintained because the central nervous system normally utilizes only glucose for energy. The sources of blood glucose during the post-absorptive state are firstly glycogenolysis, or the breakdown of glycogen in the liver. This is the first line of defense against falling blood glucose levels, but it can only provide the body's glucose requirements for 2 to 3 hours. Muscle glycogen also contributes to the maintenance of blood glucose, but it does so indirectly via the liver's processing of lactate. The catabolism of triglycerides in adipose tissue yields glycerol and fatty acids, a process termed lipolysis. The glycerol and fatty acids then enter the blood by diffusion. The glycerol reaches the liver and is used to synthesize glucose. A few hours into the post-absorptive state, protein becomes another source of blood glucose. Protein breakdown can supply large quantities of amino acids which enter the blood and are taken up by the liver, where some can be metabolized via the alpha-keto pathway to glucose. This glucose is then released into the blood. Synthesis of glucose from such precursors as amino acids and glycerol is known as gluconeogenesis, that is, creation of new glucose. During a 24-hour fast, gluconeogenesis provides approximately 180 grams of glucose. Finally, during the post-absorptive stage, most organs and tissues other than those of the nervous system significantly decrease their glucose catabolism and increase their fat utilization. This metabolic adjustment, known as glucose sparing, spares the glucose produced by the liver for the use of the nervous system. The liver can also convert fatty acids to ketones, which provide an important energy source during prolonged fasting for many tissues, including those of the nervous system. Now, while it's important that you understand how the different fuels move into and out of tissues and cells during the two metabolic phases, I appreciate that these are complex pathways to visualize. So I've included a summary table. Now, this table reiterates that glucose is the primary fuel for ATP production in all tissues during the absorptive phase, with any excess glucose converted to glycogen in the liver and muscle and triglycerides in fat. In contrast, in the post-absorptive phase, glucose produced by glycogenolysis and gluconeogenesis in the liver is almost exclusively used by the central nervous system, with other tissues switching to other fuel sources for ATP production. Okay, that brings us to the end of this presentation. You have learnt a lot about the fate of fuels in the absorptive and post-absorptive phases. It's important that you understand these basic concepts, as we will touch on them throughout the endocrine, digestion and metabolism components of this unit. With that in mind, have a go at the activity below to help consolidate your knowledge. And in the next presentation, we will focus on the hormone that regulates the transition between the absorptive and post-absorptive states, insulin. I'll see you then.